Hello friends, welcome to my new video, and in this lecture we will discuss about the smooth muscle. And learning objective of today's lectures is electrical activity in single unit smooth muscle and multi-unit smooth muscle fibers, contractile process, neuromuscular junction and control of smooth muscle and latch bridge mechanism. Electrical activity in single unit smooth muscle. So usually 30 to 40 smooth muscle fibers are simultaneously depolarized, and which leads to development of self-propagating action potential. And it is possible because of gap junction and syncytial arrangement of the single unit smooth muscle fibers. So in a single unit smooth muscle fibers, the gap junctions are present. So because of the presence of the gap junction, action potentials are transmitted very fast from one cell to another cells, so that it acts as a syncytium. Then about the resting membrane potential. So resting membrane potential in the visceral smooth muscle fiber is very unstable, and it ranges between -50 and -75 millivolt, and sometimes it reaches the low level of -25 millivolt. So in this figure, resting membrane potential in visceral smooth muscle, and it is a fluctuating type, slow wave type. Cause for the unstable resting membrane potential. So unstable resting membrane potential is caused by the appearance of the some wave-like fluctuation called slow waves. The slow waves occur in rhythmic fashion at a frequency of about 4 to 10 per minute with the amplitude of about 10 to 15 millivolt, and the cause of the slow wave rhythm is not known. It is suggested that it may be due to the rhythmic modulation in the activity of the sodium potassium pump. The slow wave is not action potential, and it cannot cause the contraction of the muscle, but it initiates the action potential. Now, three types of the action potential occur in the visceral smooth muscle fibers. Spike potential initiated by the slow wave rhythm, and action potential with plateau. So first of all, spike potential. Spike potential in the visceral smooth muscle appears similar to that of the skeletal muscle. However, it is different from the spike potential in skeletal muscle in many ways. In smooth muscle, the average duration of the spike potential varies between 30 and 50 milliseconds. And its amplitude is very low, and it does not reach the isoelectric base. Sometimes the spike potential rises above the isoelectric base, and that is known as overshoot. And the spike potential is due to nervous and other stimuli, and it leads to contraction of the muscle. So this is a figure of spike potential. Spike potential initiated by slow wave rhythm. Sometimes the slow wave rhythm of resting membrane potential initiates the spike potential, which lead to contraction of muscle. The spike potential appear rhythmically at a rate of about 1 or 2 spikes at the peak. These are a spike. 1 or 2 spikes at the peak of each slow wave. The spike potential initiated by the slow wave rhythm cause rhythmic contraction of the smooth muscle. This type of the potentials appear mostly in the smooth muscles, which are self-excitatory and contract themselves without any external stimuli. So the spike potential initiated by the slow wave rhythm are also known as pacemaker waves. The smooth muscles showing the rhythmic contraction are present in some of the visceral organs such as intestine. Then action potential with plateau. So this is a plateau. This type of the action potential starts with the rapid depolarization as in the case of the skeletal muscle. But the repolarization does not occur immediately. The muscle remains depolarized for long periods of about 100 to 1000 milliseconds. And this type of the action potential is responsible for sustained contraction of the smooth muscle fiber. After the long depolarized state, the slow repolarization occurs. So this is a slow wave rhythm of resting membrane potential. This is a spike potential, and spike potential initiated by the slow wave rhythm, and this is a action potential with plateau. Tonic contraction of the smooth muscle without action potential. Now the smooth muscles of the some visceral organ maintain a state of the partial contraction known as tonus or tone, and it is due to tonic contraction of the muscle that occur without any action potential or any stimulus. Sometimes the tonic contraction occurs due to the action of the some hormones. Then what are the ionic bases of the action potential? The important difference between the action potential in the skeletal muscle and smooth muscle lies in the ionic basis of depolarization. In the skeletal muscle, the depolarization occurs due to opening of the sodium channel and entry of the sodium ions from the extracellular fluid into the muscle fiber. But in the smooth muscle fiber, the depolarization is due to entry of the calcium ion rather than the sodium ion. Unlike the fast sodium channel, the calcium channels open and close very slowly, and it is responsible for the prolonged action potential with plateau in the smooth muscle. The calcium ions plays a very important role during the contraction of the muscle. Then electrical activity in multi-unit smooth muscle. Electrical activity in multi-unit smooth muscle is different from that in the single unit smooth muscle. Electrical changes leading to contraction of the multi-unit smooth muscles that are triggered by the nervous stimuli. The neurotransmitter depolarize the membrane of the smooth muscle fiber, slightly leading to contraction, and the action potential does not develop. This type of the depolarization is known as local depolarization or excitatory junctional potential. This local depolarization travels throughout the entire smooth muscle fiber and cause contraction. Local depolarization is developed because the multi-unit smooth muscle fibers are too small to develop the action potential. Then contractile process in smooth muscle. Compared to the skeletal muscle, in smooth muscle the contraction and relaxation process are very slow. The latent period is also long, and the total twitch period is very long, and it is about 1 to 3 seconds. In the skeletal muscle, the total twitch period that is about 0.1 second. Then molecular basis of the smooth muscle contraction. The process of the excitation contraction is very slow in smooth muscle because of poor development of L-tubule sarcoplasmic reticulum. So the calcium ions which are responsible for the excitation contraction coupling must be obtained from the extracellular fluid. It makes the process of the excitation contraction coupling slow. Now calcium calmodulin complex. Stimulation of the ATPase activity of the myosin in smooth muscle is different from that in the skeletal muscle. In smooth muscle, the myosin has to be phosphorylated for the activation of the myosin ATPase. So phosphorylation of the myosin occurs in the following manner. First, calcium that enters the sarcoplasm from the extracellular fluid that combines with a protein that is known as calmodulin and forms a calcium calmodulin complex. Now this calcium calmodulin complex that activates calmodulin dependent myosin light chain kinase. This enzyme in turn causes phosphorylation of the myosin followed by activation of the myosin ATPase. Now the sliding of the actin filaments starts. Phosphorylated myosin gets attached to the actin molecule for longer period, and that is known as latch bridge mechanism, and it is responsible for the sustained contraction of the muscle with expenditure of the little energy. Relaxation of the muscle occurs due to dissociation of the calcium calmodulin complex. So this is a flow chart for the muscular contraction or molecular basis of smooth muscle contraction. Calcium calmodulin complex is formed and that activates the myosin light chain kinase, and there is a phosphorylation of the myosin and then activation of the myosin ATPase and then muscular contraction. So this is this figure that shows the mechanism of the smooth muscle contraction. So first of all, calcium. So intracellular calcium concentration increase when calcium enters the cell and it is released from the sarcoplasmic reticulum. Now this calcium that binds to calmodulin. This calcium binds with the calmodulin and calcium calmodulin complex form. Now this calcium calmodulin complex that activates the myosin light chain kinase, and this myosin light chain kinase phosphorylate light chain in the myosin head. Okay. So myosin light chain kinase that phosphorylates light chain in the myosin head and increases the myosin ATPase activity. Now this active myosin cross bridges slide along the active actin and creates a muscle tension. So increased muscle tension. Then mechanism of the smooth muscle relaxation. So free calcium in the cytosol decreases when the calcium is pumped out of the cell or back into the sarcoplasmic reticulum. Now the calcium unbinds from the calmodulin. Then myosin phosphatase removes the phosphate from the myosin and which decreases the myosin ATPase activity. And less myosin ATPase results in a decreased muscle tension. So this is a figure of both smooth muscle contraction and smooth muscle relaxation. So this one is a relaxed smooth muscle and this one is a contracted smooth muscle, and that is known as latch bridge mechanism. So here you can see this is a thick filaments, thin filaments, and this is a plasma membrane and this is a dense body. So this is a position of thick and thin filaments in a relaxed smooth muscle and here in contracted state. Then what is plasticity? Length tension relationship. Plasticity is the adaptability of the smooth muscle fibers to a wide range of lengths. If the smooth muscle fiber is stretched, it adapts to this new length and contracts when stimulated. Because of this property, the tension produced in the muscle fiber is not directly proportional to the resting length of the muscle fiber. In other words, the Starling law is not applicable to smooth muscle. So Starling's law that is applicable in the skeletal muscle and cardiac muscle, and the tension or force of the contraction is directly proportional to the initial length of the muscle fiber in these muscles. The property of the plasticity in smooth muscle fiber is especially important in digestive organs such as stomach, which undergoes remarkable changes in the volume. In spite of the plasticity, smooth muscle fibers contract powerfully like the skeletal muscle fibers. And smooth muscle fibers also show sustained tetanic contraction like the skeletal muscle fibers. Neuromuscular junction in smooth muscle. So well-defined neuromuscular junction are absent in the smooth muscle. The nerve fiber do not end in the form of the end plate like the skeletal muscle. Instead, these nerve fibers ends on the smooth muscle fibers in three different ways. First, the nerve fibers diffuse on the seat of the smooth muscle fibers without making any direct contact with the muscle. The diffuse nerve fibers form diffuse junction. which contain neurotransmitter. Neurotransmitter are then released into the matrix which coats the smooth muscle fiber. And from here the neurotransmitters enter the muscle fibers. And in some smooth muscle fibers, the axon terminal ends in form of many varicosities. So these are a varicosities. The varicosities have vesicles which contain neurotransmitters. So this is a varicosities and it contains a vesicles containing neurotransmitters, and these are the mitochondria. Neurotransmitter is released from the varicosities through their wall into the muscle fiber. In some of the multi-unit smooth muscle fibers, gap junction is present between the varicosities and the membrane of the smooth muscle fibers, which resembles like the synaptic cleft in skeletal muscle. And the width of this gap is 30 to 40 nm, and this gap is called contact junction, and it functions as a neuromuscular junction of skeletal muscle. So this is a nucleus and this is a gap junction, and these are a dense body and dense plaque. So thick filament, these are a thin filament, and this is a smooth muscle cell. So dense plaque couple adjacent cells mechanically, and these are the gap junction couple the adjacent cells electrically and chemically in some smooth muscle types. Then control of the smooth muscle. Smooth muscle fibers are controlled by nervous factor and humoral factors. So nervous factors, smooth muscles are supplied by both sympathetic and parasympathetic nerve, which antagonize each other and control the activities of the smooth muscles. However, these nerves are not responsible for the initiation of any activity in smooth muscle, and the tone of the smooth muscle is also independent of the nervous control. Then humoral factors. Activity of the smooth muscle is also controlled by humoral factors, which include hormone, neurotransmitters and other humoral factors. Now hormones and neurotransmitters. Action of the hormone and neurotransmitter depends upon the type of the receptors present in the membrane of the smooth muscle fibers in a particular area. The receptors are of two types, excitatory and inhibitory receptors. If excitatory receptors are present, the hormones or neurotransmitters contract the muscle by producing depolarization. And if inhibitory receptors are present, the hormones or neurotransmitter relax the muscle by producing hyperpolarization. Then hormones and neurotransmitters which act on smooth muscles are acetylcholine, antidiuretic hormone, adrenaline, angiotensin 2, 3 and 4, endothelin, histamine, noradrenaline, oxytocin, serotonin, and other humoral factors other than the hormones that cause relaxation of the smooth muscle fibers. And the humoral factors which relax the smooth muscles are lack of oxygen, excess of carbon dioxide, increase in hydrogen ion concentration, adenosine, lactic acid, then excess of potassium ion, decrease in calcium ion, nitric oxide, endothelium derived relaxing factor. Thank you.